All right, in review, mitosis is what we talked about before, and that's where body cells are going to try to divide and make more exactly like it. So these are identical at the end. Identical. So what I want to do now is focus more on these gametes, the sperm and the egg. So we see here, this egg only has one set of chromosomes and the sperm does too, because they are two halves that have to come together to make a whole. So if body cells make identical cells like this, how are these made? How would sperm and egg made? It must be a little bit different, and it is. So that's what we're going to focus on now. The germ cells is what they're called. They are the parent cells that divide to make sperm or egg. They do a division that's just for them. It's called meiosis. Look at the spelling there, Meiosis. So meiosis is just for the formation of sex cells. Here's what they do. They start out in interphase, just like you noticed before. Look at what the chromosomes look like at this point. I'll wad it up just like before. We're going to call that form of chromosomes chromatin, just so we can kind of keep track of what those chromosomes are doing. So chromatin is kind of all wadded up in that nucleus. When prophase starts, they're going to break out. You see these X's that are forming. They don't look all wadded up, and the nucleus is starting to dissolve. In meiosis, we have a 1 or a 2 that's after the number. So you do have to write that number. Like prophase 1 is the first thing. So the X's are showing up. This is important that happens during prophase, crossing over. That's a vocab word we need to know. This is why every time your parents have a child, they don't all look exactly alike. Because the chromosomes do this really cool dance, is what I like to think of it. The red chromosome would be the maternal, the mom chromosomes, and then the dads get really close together, so close that they form this tetrad. So each pair gets together and forms this tetrad so that some of their genetic material gets switched out from each other. This crossing over is what achieves genetic variety for the offspring. That's important. So after it does that, it's going to line up. That tetrad stays together. There they are. The whole tetrad is lined up here in the middle, and here's another tetrad here. The spindle fibers are forming from the centrioles, the end zone. They're attached to the chromosomes because next they are going to pull them apart. There's anaphase 1. See here, the tetrad has been pulled apart. You got X going over here, you got X going over there, so that by the end in telophase, you have the cell starting to pinch in half, giving us a 2 cells at the end after cytokinesis pulls them apart. The only thing is that these cells are still diploid. You still see the X's that are kind of wadded up in here inside the nucleus. That is why they have to divide again. So you're going to do all that stuff over again. Prophase 2, they break out of the nucleus. You see the X's showing up. So now the big clue to pick out section 2 is that there's 2 cells that look just alike, and they're both doing the same thing. Metaphase 2, now you have the single X's lining up in the middle. You get the spindle fibers forming here. And anaphase, they're going to pull them apart. So now they look like little V's. These V's, I'm going to call those chromatid. Chromatid, that's like half of a chromosome, this little V-shaped thing. So now they're chromatids. And then you can kind of imagine what's going to happen here. The center starts pinching off so that by the end, when cytokinesis pulls them apart, we have 4 cells that have these little half chromatids in them. So these are 4 haploid cells that are formed by the end of meiosis, and each one of them are different from the other. So they're very kind of like snowflakes. Every sperm and every egg is very different from each other. So you're going to use this to practice identifying phases. Like, for example, this one kind of has these little tetrads formed together. So that would be metaphase 1. And this one has them pulled apart. So this must be anaphase. This must be telophase because they both look the same. Telophase 1, got prophase 1 here. And you can kind of practice naming these here. I see 4, so that must be telophase 2, prophase 2,